All right, let's jump right into this explainer. We're looking at the exact thermodynamic tipping point that triggers a massive biological domino effect in marine ecosystems. We hear about temperature stress all the time, but the data actually points to something insanely precise happening just under the surface. Especially in closed systems like reef aquaria, survival isn't just about keeping things warm. It's dictated by a ridiculously rigid physical threshold. And that tragic number? 28.5 degrees Celsius. This isn't some gradual, fuzzy transition zone. It's a hard, physical pivot point where the fundamental physics of water literally betrays the biology of the organisms living in it. Below this number, everything, metabolic processes, enzymatic rates, biochemical diffusion, is beautifully balanced. But the literal second we cross 28.5, the rules of the entire ecosystem just completely flip. To really understand why this happens, we have to look at the foundation of the disaster, which is oxygen. As seawater gets hotter and the kinetic energy of the water molecules increases, dissolved gases are violently forced out of solution. By the time water hits 30 degrees, oxygen solubility drops by a massive 15.3% compared to standard keeping conditions. And the absolute tragedy here is the timing. This systemic deoxygenation happens at the exact moment that the metabolic demand of every aerobic organism in that water, the fish, the corals, the microbes, is exponentially skyrocketing because of the heat. The ratio of oxygen supply to demand simply collapses.
So what happens next? Let's get into the cellular solar meltdown. This kicks off a catastrophic failure in the coral's internal solar-powered factory, ending with the host desperately evicting its own algal workers just to survive. It starts when the combo of thermal energy and solar light damages the thylakoid membranes of the symbiotic algae, specifically knocking out a crucial protein in photosystem 2. Suddenly, the electron transport chain breaks down, but the solar energy just keeps pouring in. Since this energy can't be used for carbon fixation anymore, it reacts with ground-state oxygen to generate a massive, highly toxic wave of reactive oxygen species, or ROS. These dangerous molecules leak out of the algae, completely flooding the coral host's cells and totally overwhelming its natural antioxidant defenses. Facing massive damage to its lipids and DNA, the coral literally has no choice. It initiates programmed cell death and violently expels the damaged algae. And that eviction is exactly what we see as coral bleaching.
But here's a wild twist. Right before turning stark white, some corals go through this bizarre phenomenon called colorful bleaching. As they lose their internal photosynthetic pigments, they suddenly upregulate their own fluorescent proteins, pumping out these brilliant, vibrant neon blues, purples, or pinks. Now, this isn't just some pretty death throw display. It's actually a highly functional defense mechanism. These neon proteins act like a chemical optical sunscreen. They literally shield whatever surviving algae they have left from incoming solar radiation, basically buying themselves just a little more time in case the water cools down. Let's look at how they try to adapt, shuffling symbionts for survival. When their primary algae fails, the coral is forced into a desperate biological trade-off. It swaps out its native, highly efficient but thermally fragile symbiont called Cladocopium for a backup population of heat-proof algae called Duraverdinium trenchii. This resilient backup can survive temperatures well over 30 degrees, thanks to some unique genome duplications and tough photoprotective mechanisms. And hey, it stops the coral from starving to death immediately. But there's a huge catch. Because this backup algae is kind of a biological opportunist, it transfers significantly fewer autotrophic nutrients, like glucose and glycerol, back to the host. So sure, the coral survives the heat wave, but it's left in a severe long-term energy deficit. And because of this massive energy deficit, the host can't power the calcium ATPase pumps it needs to move protons out of its internal calcifying fluid. Without that biological energy, skeleton building just hits a brick wall. In fact, long-term core analyses show a staggering 21.8% decline in the net calcification rate of modern corals during our current epoch compared to historical baselines. They're forced to prioritize keeping their existing skeletons thick rather than growing outward, simply because they literally do not have the metabolic fuel left to do both. Okay, moving on to the unseen battleground, the microscopic bacterial war. Because crossing that 28.5-degree threshold doesn't just mess with the algae, it kicks off an all-out microscopic war on the coral's surface. Normally, corals have this amazing probiotic shield made of beneficial bacteria called Endozoicomonas. Under healthy conditions, these good microbes actually convert host cholesterol into steroid hormones, stuff like testosterone and progesterone, which provide essential antioxidant support and prime the coral's immune system. But when temperatures sustain above 28.5 degrees, these beneficial populations completely collapse. And the absolute second that shield drops, opportunistic pathogens, specifically from the genus Vibrio, flood into the vacuum and completely take over the ecosystem, causing rapid tissue necrosis. And the way these villainous Vibrio bacteria orchestrate their attack is honestly terrifying. Right above 28.5 degrees, a genetic switch flips in them. They upregulate specific proteins in pili to physically stick themselves to the host's tissue. Then they deploy these crazy syringe-like nanomachines called type 6 secretion systems. First, they use the T6S1 nanomachine to assassinate any remaining beneficial microbes, physically securing their territory. Once the coast is clear, they fire a second system, T6S2, directly into the coral host's cells, injecting up to 9 distinct lethal toxins that literally dissolve the coral tissue from the inside out. So if you're an aquarist watching this microscopic massacre rip through your captive reef, you have to respond aggressively. First step, immediately drop the water temperature below 25 degrees to slow down the pathogen's virulence. You also have to instantly cut off any organic nutrient dosing, stuff like amino acids, because that's basically throwing raw fuel on the Vibrio fire. And surprisingly enough, aquarists can even use targeted dark cycle treatments of natural witch hazel. The tannins in witch hazel physically bind to the membranes of these attacking bacterial pathogens, essentially tying them up so they can't adhere to the coral tissue.
Let's move up the food chain a bit and look at shrinking fish and brittle bones. Remember, fish are ectotherms, meaning their body temperature and their metabolic rate is entirely at the mercy of the surrounding water. So when things get too warm, a clownfish's basic energetic maintenance costs just skyrocket. Right at the exact moment, the physical oxygen in the water is plunging. To conserve their rapidly shrinking aerobic scope, they're forced into some wildly desperate adaptations. Their brains actually upregulate specific neuropeptides that suppress their own appetite, drastically reducing their food intake by 17.9% just to avoid the energetic costs of digestion. They accelerate their gut transit to prevent metabolic congestion, and incredibly, they even undergo phenotypic shrinking. Yeah, they literally physically shorten their overall body length. And amazingly, this bizarre, drastic downsizing actually works. In a desperate bid to lower their total oxygen demand and maintain social cohesion within their host anemones, shrinking their body mass increases a clownfish's survival probability during severe thermal stress by up to 78%. It's just wild. A literal physical downsizing purely to balance an impossible biological oxygen budget. But let's add some nuance here and look at regional adaptations. Because, not every marine organism is destined to instantly perish the second the thermometer ticks past 28.5 degrees. Genetic and regional adaptation is very real. If we look at wild geographic variations, corals adapted to the cooler subtropical waters of Bermuda will severely bleach right at that 28-degree mark. But check out the northern Red Sea. Corals there have a much wider safety margin, surviving up to 32 degrees. And remarkably, the highly adapted corals living in the extreme environment of the Arabian Gulf can tolerate staggering summer water temperatures up to 35 degrees before this physiological cascade even begins. Their genetic history completely shapes their resilience. The incredibly cool part is, we can actually leverage this historical resilience in captive environments through a technique called stress hardening. Studies show that if we intentionally expose corals, like Caribbean staghorn fragments, to fluctuating daily temperatures, swinging them safely between 28 and 31 degrees, we can literally rewrite their biological response. When these preconditioned corals are later hit with a sudden, acute 32-degree heat shock, they don't suffer the rapid tissue sloughing and immediate necrosis you see in control groups. Instead, that sudden death sentence is transformed into a much slower, gradual, and ultimately survivable bleaching event. So I'll leave you with this thought to ponder as we wrap up this explainer. We've just seen the immense, intricate biological warfare and structural collapse that happens within a controlled captive reef tank when the temperature edges just a fraction of a degree over that 28.5 limit. If a mere half degree triggers this unstoppable thermodynamic and microbial domino effect in our highly monitored glass boxes, what does that mean for the vast wild oceans operating right on the brink of those exact same temperatures today? It's something that definitely challenges not just how we keep aquariums, but how we view the fragility of the entire ocean ecosystem.